I'll borrow Saskia's comment that it's method day and not results day, so I'll also talk about how I'm dealing with analyzing co-registered EEG and MEG and eye tracking data and no results, sorry, so just methods. So why I'm presenting this today is that traditionally in EEG and MEG, we ask our participants to keep their eyes still, and this is because the eyes cause a lot of noise in EEG and MEG data. And this is because the eyes can be thought of as dipoles, so as they move, also these dipoles move and affect especially frontal electrodes, both in EEG but also in MEG. I'm interested in reading, so when we read, we typically made a lot of rightward saccades, and this is how they look like. So we have relatively positive electrodes on the frontal right hemisphere and negative voltages on the left side at frontal electrodes. And this is how saccades look like in terms of waveforms and times and voltages. However, this is not the only type of artifact that we get when looking at including eye movement in EEG and MEG, and we also get other type of artifacts such as spike potentials, which look very different in terms both of the waveform and also in terms of their scalp topography. So yes, we do have some artifact localized in frontal electrodes, but it's much more widespread also on parietal sides. And because EOGs, which are special electrodes that are supposed to capture movements of the eyes, do not capture this artifact very well. It's the most difficult artifact to correct in EEG and MEG data. And finally, we also have blinks and eye-related artifacts that we also need to take into account, although these are quite easier to recognize and to remove. So despite having all these problems when letting our participants moving their eyes in our paradigm, we think it's still worth it giving it a try and try to correct for these artifacts. And this is because letting our participants moving their eyes can open up a whole different type of paradigms, and it also has the huge benefit of making it possible to analyze brain and behavior at the same time. So we can both look at the brain activity that's generated by different fixations. For example, when looking at different parts of the screen, we can lock our analysis to that event and analyze the evoked response. But we can also analyze behavior in terms of saccade durations or fixation durations, saccade amplitude, and all information derived from the eye tracker. And again, I'm interested in reading, so especially for reading, the traditional approach is to present words serially instead of presenting whole sentences. So we present one word at the center of the screen and then the following word, so that the participants don't have to move their eyes but can always look at the center of the screen and passively read. However, reading is not as passive as assumed in serial visual presentation. Indeed, there are skips, which means that sometimes we don't look at a word, we regress to previous part of the sentence, and especially we use information from parafoveal vision, which means that we don't only analyze the word that we are actually looking at, but we also process words that will be further down the sentence. And for all these reasons now, co-registering, eye tracking, and EEG is becoming more common. There are now a number of studies available using this technique. However, the further inclusion of MEG is much more rare, and there are only a few studies looking at reading in combining EEG and MEG, eye tracking and MEG. And there's just one study that's been done here at the CBU, more a proof of concept study that's combining all of these together. So to get a bit more concrete, the first thing to consider starts before analyzing the data, and it starts when preparing the presentation of the stimuli. And it's important to remind that we now have two streams of data, the MEG and the eye tracking data, and they are independent of each other, so they work on different timelines. And in order to make sure that we can use information from the eye tracker in order to analyze brain response, we need to make sure to send triggers that are as synchronous as possible, so to send triggers at the start and at the end of each trial, so to be able to then align them and import information from the eye tracker to the MAC data, so that we know when participants are looking at, if it's a visual experiment, where participants are looking at, and use that information to study evoke responses. In this case, it will not be any more ERPs or ERFs. It will be fixation-related potentials and fields. And to be a bit more concrete, if I'm presenting some sentences to participants, they will read a sentence basically jumping with their eyes from one word to the next. And there might be a target word that we are interested in, and we, using information from the eye tracker, we can know exactly when they look at that word to generate fixation-related fields in this case. So this looks like a reasonably good visual evoked field. So these are magnetometers. We see that there's a P1. There's still maybe some noise here due to saccades, and it gets a bit noisier as we go further down the epoch, but it looks like a traditional ERF. So again, the big problem is how to remove these ocular artifacts, which are necessarily there when allowing our participants to move their eyes. And for doing this, we are going. This is exactly where I am at now in the process of analyzing this data. So we are trying to use a traditional approach of using ICA to determine, to fit the data with and decompose our signal in different components. And the idea is to determine which components correlate with eye movements and remove those components from the data, so to have just brain signal, or just not eye movement artifacts. However, we need to be a bit more careful than in traditional settings. So we need to be a bit more careful to ensure that the data the ICA is fit with contains enough noise and signal, and we need to check that the ICA is actually stable, so that there are only a few components that relate to all the ocular artifacts and only the ocular artifacts, and then to be able to remove those components. For this work, we are using some guidance that's out there for co-registration of EEG and eye tracking, and we are just following their suggestions and trying to apply different filters, both high pass and low pass through the data. And importantly, a suggestion that's been very useful is to overweight saccade events, which just means to oversample those parts of the data that contain a saccade, so that there will be relatively more saccade than there actually is, to make sure that the ICA is able to pick up especially the spike potential, which is the most difficult to correct. And another difference compared to traditional ERP designs is to use a different approach for selecting ICA components. So traditionally, we correlate all the components with the two EOG's channels, and this would give us two components to remove from the whole array of data. But it's been suggested that it's more, since there will be a lot of eye movements, and the EOGs do not pick up especially spike potential, to use this another method of looking at the variance of the signal of the components during saccade and during fixation events, which basically just means that the assumption is that during saccade events, components that relate to eye movements will be more active. So there will be a higher mean variance during saccade events than during fixation events. And in this way, we don't get just the two components that we get with the traditional approach, but we get also another components which is related to the spike potential. And let's look at some data. Okay, so this is, let's see if I can increase this, yes. So this is how the fixation-related potentials look like when uncorrected. So we see that there's this spike potential. This is a nice evoked response with a P1. And then when, however, there's still the corneoretinal dipole and the spike potential. And this is for EEG, which are the mostly affected by eye movements. And there's still noise in radiometers and magnetometers. And we tried a few different pipelines for correcting for all these artifacts. This is a standard ICA approach, and we see that the spike potential is still there, is in magnitudes is quite as big as a P1 response. So we tried a lot of different things and pipelines, and for this participant we were able to get a nice and clean signal, especially for EEG. For radiometers and magnetometers also. This is how it looks like. However, unfortunately, we have not come up with a full pipeline yet, because what works for some participant does not work for others. So for this participant, for example, by applying the same pipeline, the data basically don't change that much. We still are in both the corneoretinal dipole and the spike potential are still present in the data. So if anybody of you has worked on similar type of problems, I'm very happy to talk and hear other suggestions on other possibilities on how to clean this type of data with ICA. And let me resume. Good, so I'd like to thank the people I've been working with on this project, which are Olaf and Prane here at the CBU. And before finishing, I'm still collecting data. So if you want me, if you can help me finish before Christmas, I'd really appreciate it. Thank you.